This procedure begins with culturing the bait strain in media containing normal amino acids and the silac control strain in heavy isotopic lysine and arginine. Equal amounts of both strains are mixed and lysed by grinding in liquid nitrogen. Crude lysate is first pelleted by centrifuge and then cleared by binding to cephalose beads. Cell lysate is then bound to IgG beads. The proteins bound to IgG beads are eluted by protease cleavage and precipitated. The sample is now ready for analysis by mass spectrometry.
Hi, I'm Jesse Chow from the Lone Lab in the Department of Cell and Developmental Biology at the Life Sciences Institute of the University of British Columbia. Today we will show you a procedure for quantitative proteomic analysis called CyLac combined with affinity purification. We use this procedure in our laboratory to study protein complexes. So let's get started.
In this procedure, the bait strain contains normal amino acids while the silac control strain contains heavy isotopic lysine and arginine. To get started, first grow 1 liter each of the bait strain and the control strain separately to the values shown on your screen. Next, pour the culture into 500 milliliter Nalgene centrifuge tubes. Balance the load in pellet cells at 2,580 RCF. Decant the media and resuspend the pellet with 50 milliliters of cold DH2O while transferring the cells to 50 milliliter centrifuge tubes. Centrifuge the tubes to pellet the cells at 3000 RCF. At this point, you can freeze the pellet at -80 degrees Celsius. It is important to remember to match the control strain culture to the bait strain culture in 1:1 by dry pellet weight. Resuspend each cell pellet in 10 milliliters of NP40 buffer with a 1 to 2000 protease inhibitor cocktail. Mix the two cell cultures by directly decanting one tube to the other. To begin grinding the cells, first pour liquid nitrogen into the pre-chilled mortar and allow it to completely evaporate. Next, add 2 milliliters of cells to the mortar. Pour some liquid nitrogen to freeze the cells. Grind the cells until the cells become fine powder. Repeat the process until all the cells are ground. Be sure to not allow the cells to thaw. Add liquid nitrogen when necessary. Next, transfer the powder to an ice-cold beaker and thaw at room temperature. When the edges begin to thaw, add 20 milliliters of NP40 buffer with a 1 to 200 PIC. After it has thawed, transfer the crude lysate to 40 milliliter Nalgene tubes and spin at 16,000 RPM in a Sorval SA600 rotor for 30 minutes. While waiting, take 300 microliters of the cephalose 2B beads and wash the beads in 300 microliters of NP40 buffer 3 times. Next, resuspend the beads in 300 microliters buffer so that they are in a 1:1 slurry. The next step is to pre-clear the cell lysate. To do this, first transfer the supernatant to a new tube and add the cephalose 2B beads. Incubate on a rotating platform in a cold room for 30 minutes. While incubating, take 300 microliters of IgG cephalose 6 beads and wash the beads in 300 microliters of NP40 buffer 3 times. Then resuspend the beads in 300 microliter buffer so that they are in a 1:1 slurry. After incubation, pellet the cephalose 2B beads and transfer the supernatant to a new tube. Remember to save an aliquot of the cell lysate for Western blotting later. Then, add the IgG beads. Separate the contents into 2 tubes for more efficient binding. Incubate on a rotating platform in a cold room for 2 hours. After incubation, collect the beads in a chromatography column at 4 degrees Celsius. Be sure to save an aliquot of the unbound fraction. Wash the beads with 10 ml NP40 buffer. Next, wash the beads with 3 ml TEVC buffer. Again, remember to save an aliquot of the beads. This will reflect the efficiency of binding. To elute the beads, first add 5 microliters of ACTEV to 1 milliliter of TEVC buffer. Add TEVC buffer to the beads and mix. Transfer the contents into 2 1.5 milliliter Eppendorf tubes for more efficient mixing and incubate on a rotating platform in a cold room overnight. Next, spin down the beads and transfer the eluate to a fresh 1.5 milliliter tube. Wash the beads with additional 0.5 milliliters of TEVC buffer. After washing, combine the eluate in one tube. You should have 1.5 milliliters of eluate in total. Save an aliquot of the TEV eluate. To precipitate, adjust aliquots to 25% TCA with 100% TCA. To do this, separate the 1.5 milliliter eluate into 2 tubes of 750 microliters each. Add 250 microliters of 100% TCA to the tube. Your solution should turn milky. Next, place it on ice for 30 minutes, periodically vortexing the mixture. After incubation, spin at maximum speed in the tabletop centrifuge in a cold room for 30 minutes. Wash once with 500 microliters of ice-cold acetone containing 0.05 normal hydrochloric acid. Then, spin for 5 minutes at maximum speed at 4 degrees Celsius. Next, wash once with 500 microliters of ice-cold acetone and spin for 5 minutes at a maximum speed at 4 degrees Celsius. After spinning, carefully remove the acetone and dry pellet. For silver staining, resuspend the pellet in 50 microliters of 1 times the SDS sample buffer. If you are planning to use the precipitated protein for mass spectrometry-based analysis, then it is recommended to precipitate using ethanol. To do this, first add 20 micrograms of glycogen. Next, dilute samples 5 times with 100% ethanol and adjust to 50 millimolar sodium bicarbonate with a 2.5 molar stock at pH 5.0. Mix the solution by inverting the tube. Let it stand for 2 hours at room temperature. Finally, split the contents into 2 Eppendorf tubes. Pellet the precipitated protein by centrifugation for 10 minutes at 12,000 Gs at room temperature. The aliquots saved during the purification procedure should include the pre-cleared cell lysate, the bound fraction, the unbound fraction, and the eluted fraction. We recommend analyzing the protein contents of the above aliquots by Western blotting using anti-TAP antibody or silver staining to reflect the binding and eluting efficiency of the experiment. In this silver stain gel, purified SCS2-TAP and its interacting partners were visualized and compared to an untagged control. In this Western blot, various factions taken during the purification of SCS2-TAP were analyzed by Western blotting using anti-TAP antibody. Notice that SCS2-TAP migrated at a lower molecular weight after elution by TEV protease cleavage, and there was no SCS2-TAP left on the IgG beads after elution. This confirmed that the purification was highly efficient.
We've just shown you how to purify protein complexes by affinity purification with IgG beads. When doing this procedure, it's important to remember to be sure to match the base strain and the control strain by dry pellet weight before you begin the purification procedure. Since CyLac provides us with unbiased and quantified measurements of protein binding partners, we only carry out the first stage of the TAP purification to minimize sample loss. If you experience significant amounts of unspecific binding, you may wish to carry out the entire protocol, which can be found in the Cold Spring Harbor manual. So that's it. Thanks for watching and good luck with your experiments. Hey, come on, Mark. Oh,